All right, good evening, everyone, and welcome to Strait Science. Strait Science is an evening seminar series put on by University of Alaska Fairbanks Alaska Sea Grant and University of Alaska Northwest Campus, both in Nome. Alaska Sea Grant and Northwest Campus are public servants of the Bering Strait region. We serve all peoples of the Bering Strait region, which is the homeland and waters of the Inupiaq, St. Lawrence Island Yupik, and Central Yupik peoples. Now, tonight we're doing something, we have a lot of speakers. We have a real straight science topic talking about the Bering Strait land bridge. And I'm going to do a round of introductions, and the people who are going to speak are going to wave their hand and let you know who they are. So here we go. We've got 6 speakers tonight, and they're gonna hand it off to the next person. And starting, and this is in the order of talking, we have Sarah Fowell. Sarah is a professor in the Department of Geosciences at UAF, and I'll read it all. Her research uses information from lake cores, including sediment composition, pollen, and spores, to reconstruct past climate and vegetation cover. Second, and then maybe Sarah, if you turn off your screen, we'll winnow it down. Second is Beth Caissie. Beth is a research geologist at the USGS, U.S. Geological Survey, in California. Her research looks at how the climate of the oceans, particularly the Bering Sea, has changed over the past tens of thousands of years to hundreds of thousands of years. So that's we could use you now in the past and in the future. Jenna Hill, there you go. Research geologist, also at the USGS in Santa Cruz. Her research looks at acoustic data from the ocean floor to reconstruct ancient landscapes and depositional environments. So she's all about the mud. Oh, I've sort of goofed this up. I'm sorry. Jenna will go before Beth. Then Joshua Barnes. Joshua, unmask yourself. Josh is a PhD student at UAF, and his research focuses on the use of pollen for vegetation reconstructions of the emergent Bering land bridge. And he's figuring out how the landscape may have affected megafaunal migration, so the big animals, between North America and Eurasia. Ryan Ostie, nope. Kira Wankett, down there, yep, I see you, great. Kira is a graduate student at the University of California, Santa Cruz. She uses sedimentary ancient DNA to investigate ice age plant and animal communities. That sounds really cool. And bringing it all home, and he will bring it all together at the end, is Ryan Ostie. Ryan is a PhD student at the University of Alaska Fairbanks, studying Earth systems science. Their research focuses on how past ocean and atmospheric climate changes have been recorded within the geological record. So we have a lot going on, and they're going to focus tonight. Now, in 2023, we had Sarah Fowell had started off, and she had presented with David Scholl and another gentleman, I think.
It was with Beth Caissie.
Beth Caissie, I'm sorry, Beth Caissie, who is on tonight. And they were headed out to take these sediment cores. So they were going to dig down and find out, sort of go back in time when the land bridge was above all the ocean we have now. And so now they're 2 years later, they're back and they're giving us an update on what they found. So we're really grateful. We live on the Bering land bridge, and so it's of great interest to the region what you found. With that, we'll turn it over to Sarah. And take it away, Sarah Fowle. We're so glad you and your team are here.
All right. Thank you, Gay. It was great to be invited to come back and tell you a bit about what we know. But I do want to stress this is very much a team effort, which is why you've got 6 presenters tonight and a work in progress. We've learned a lot of things, but we have a lot to do. What, Gay?
You can put your video on.
I will.
There you go.
I'm back.
You can see it. Okay. Thank you so
Okay,
much.
so I'm going to start by kind of presenting the Mile High overview, and then each of the other presenters will zoom in on their area of expertise. So the big question we wanted to be able to answer with this project is whether the vegetation and climate of the Bering Land Bridge were similar to those of Alaska to the east and Siberia to the west during the last ice age. And shown here, we've got an artist's reconstruction of Alaska during the last ice age. The vegetation cover was an arid Arctic grassland. And of course, that grassland supported a lot of grazers, including horses and mammoths and bison. So were the lowlands of the Bering Land Bridge similar to this vast arid grassland or different? And that question is difficult to answer because the land bridge was flooded as glaciers melted and we came out of the ice age, sea level rose, and now most of that land bridge lies below sea level and below layers of marine sediment. So that makes it difficult to sample. Come on, you can do it. There we go. So in August 2023, we sought to sample sediment deposited on lakes and ponds on the emergent Bering Land Bridge when it was above sea level in order to answer some of these questions that you see on the right side of the screen. And really the big question was what was growing on the land bridge, what lived on the land bridge? What was it like? We had initially planned to target some sedimentary basins, sediment-filled low spots. We thought if these were low spots that were subsiding, they would have been low areas on the land bridge. They might have then filled with water and housed several large lakes. And there's sort of these blue dashed outlines here. There's one here, there's one here, one over here, and a couple down here. And that kind of gave us our original cruise plan for where to sample. Well, we found something a little bit different than we expected. We did find lakes, but instead of several relatively large lakes, we found evidence for many smaller water bodies between old river channels out there on the Bering Sea shelf. We successfully retrieved freshwater sediment deposited in ponds or bogs on the Bering Land Bridge from 28 of the 30 4 coring sites that are shown here. These are the ones, the yellow dots on this map, and this is a map of sites that we cored where we retrieved sediment cores. These yellow dots went all the way through the marine sediment into sediment deposited in freshwater bodies on the Bering Land Bridge. And those can help us answer questions such as some of these over here on the right. What plants were present on the Bering Land Bridge? How did changes in latitude, elevation, and distance from the coast affect those plant communities? And what resources were available for animals on the land bridge? 6 of these coring sites, the ones that are sort of teal colored or blue-green dots on this map, are entirely marine, and that was intentional. They come from deeper water, and we were hoping that those cores would help us answer questions like this one. Is there a correlation between sea ice extent in the Bering Sea and arid conditions on the Bering Land Bridge? Central Varangia is another name for that region. So, come on, you. Between early August and early September of 2023, we used the research vessel Sikuliak to identify buried basins on the Bering Sea shelf and core them. Coring sites were identified using the Sikuliak sub-bottom profiler. There we were looking for buried basins, and you can see these kind of bowl-shaped features beneath relatively flat-lying marine sediment. Sometimes they're more U-shaped features, but we're looking for these bowl-shaped features with layers of identified layers of sediment below the relatively flat-lying marine blanket. And we didn't
Quick question, Sarah.
absolutely.
When
Yes?
we're looking at your picture here, Is
Yes.
that the ocean seafloor? That's the
This is
dark
sediment
line?
on the seafloor. So
Okay,
this one, and Jenna's going to talk more about this in a minute, but this sort of layers on top, that's the marine sediment. Stuff below, like this thing is a buried basin.
okay, but it's just a picture, like a it's a, like if you're cut into the sediment there with your,
Yeah, it's yes, you can think of it that way.
yeah,
And we're
thank
using
you.
a sub-bottom profiler, which acoustically images layers of sediment on the seafloor.
Thank you.
So once we identified these buried basins, we had a couple different types of cores at our disposal. A lot of our data was collected using a gravity core, which has this long tube attached to the coring head. You can put weights in here, and then it's lowered quickly to the seafloor on a cable. It sinks into soft mud under its own weight, and you can pull it out and therefore collect cores. In places where the sediment was stiffer, sometimes if you've got a thick sandy layer, the gravity core won't be able to go through it. And then we use a vibrocore, which actually has this box on top, which shakes the coring tube just a little bit. It vibrates it so that it can liquefy sandy layers and hopefully then sink down through some of those layers that were a little too stiff for the gravity core. And we collected many of both types of cores. We opened and described some of the cores on board. We just wanted to make sure we were finding what we were looking for, that those basins really were freshwater ponds or bogs or lakes. And this is kind of a pretty good example of what we often found. This is a top part of this core, and it's relatively boring looking gray-green marine mud. But as you go down the core, you start to get into this nicely laminated sediment with layers that are pretty rich in organic matter. And those we have determined were deposited in a freshwater setting. And in order to determine, in order to figure this out, one thing we did on board the ship was take little samples of sediment, sieve them. That allowed us to pull out some tiny fossils. And from the laminated layers, we got things like this: freshwater plants and animal parts, seeds of terrestrial plants, and lots of moss leaves from sphagnum. Once we got back to the lab and back to the core lab, we were able to split and sample a lot more of our cores. We haven't done all of them yet. This is stable carbon values and carbon to nitrogen ratios from 9 different cores. And this also helped us to confirm that the lower parts of many of our cores accumulated in a freshwater setting, because the carbon isotope signature and the carbon nitrogen ratios of organic matter from terrestrial sources sort of are represented by this upper left quadrant on all these diagrams. So if the dots are in there, the dominant source of organic matter is plants or soil. Organic matter in the cores that came from a primarily marine setting is plots down here in the lower right on these diagrams, meaning that the source was primarily marine phytoplankton, marine algae. And you can see some of these cores, as designed, are entirely marine, but most of them have samples from the lower part of the core that appear to come from a mostly terrestrial source. Those we're identifying as freshwater bodies, and they have freshwater organisms in them. And then an overlying layer of sediment that comes from a marine source that was deposited on top as the land bridge flooded. And some of these cores are predominantly freshwater. And on this diagram, you can see sort of the proportions of freshwater versus marine sediment in 23 cores that we have split, described, and sampled. The freshwater part is sort of this gray-green. The marine part is the blue on top. Again, some of these are entirely marine. And if you look at the dates, we do have dates for many of these cores. Dates range from about 13,000 years, calibrated dates, 13,000 years before present for some sites. Sledge Lake site is actually in Norton Sound. We have basal dates of about 49,000 years from the Camelback site, which is farther south on the Bering Sea shelf. We are still continuing to pick material for dating. Right now, if you're looking at these, you can see some reversals where you have older ages over younger ages in the freshwater portions of these cores. We think the reversals may indicate that some of these freshwater bodies were thaw lakes. So as they thaw and expand, older materials coming out of the lake shore, or possibly they might also indicate the presence of reworked material in a floodplain setting. And we have plenty of evidence for floodplain settings. So again, we are still picking material for additional dates. We're going to be splitting, sampling, and describing additional cores in May, so in a couple of months, and more data will be coming forward over the course of the next year. And with that, I'm going to turn it over to Jenna.
All right, I'm doing my best to get the share going quickly. All right, hopefully you can see my screen.
It's perfect.
So I'm going to talk a little bit about how we pick some of the core location sites. And so as mentioned in the beginning, my background is using acoustic data and things like sonars to collect data of images of what the seafloor looks like in terms of the shape and structure of the seafloor. And then we can also use sonars to look at images from beneath the seafloor, so layers of sediment beneath the seafloor. So to do that with the RV Sikuliak, we use a system that's called a chirp sub-bottom sonar. And essentially, it's a sonar instrument that is mounted on the hull of the ship. And so there's a nice little picture. I'm going to try to switch to a laser pointer here. There's a nice little picture here that Sikuliak has, where if you were to take the ship out of the water and look at the bottom, they have a whole array of sensors that are mounted on the bottom of the vessel here. And those all use sound. So that's what a sonar is. It emits a sound. That pulse of sound goes out into the ocean. It goes down through the water column, and then it reflects off the seafloor, and some of that sound goes into the layers of sediment or rock beneath the seafloor and bounces back up to the ship. So it sends a signal back up to the ship that then we convert into these kind of sub-bottom profiles and seafloor maps. And so I'm trying to show a little example of this, so we do this ping, ping, ping constantly and use this sound to create these kind of profiles and images of the seafloor. And this is just a picture that I thought was really nice of one of the Sikuliak techs on the vessel explaining sort of how this works, and gives you also a view of what the lab looks like. There's a lot of computer screens with a lot of data coming in all at once. And so one of the nice things is that we can actually see the data as we're collecting it, and we know in real time what we're getting. So we use this sound to create some of those images that Sarah was showing you in the beginning. And let's see if my screen will advance. It is not advancing.
I was having to click on it once and then hit the arrow keys.
Click. Yeah, thank you. Okay, so we are looking, as Sarah said, for paleo channels, river valleys, lake basins. And so how do we find those using this sonar? So this is an example of a sub-bottom profile here on the bottom, and I've tried to sort of color code and give you a little bit of information about what's going on. Usually what you'll see in these profiles is a dark black line here that represents the seafloor, and then the layers that we're looking at beneath that are all layers of sediment beneath the seafloor. And so a lot of places what we'll see, as Sarah mentioned, is sort of a layer of marine sediment that was deposited after the land bridge was flooded. But the part that we're really interested in is those terrestrial sediments that are preserved and not washed away when sea level rises. So we look for these kinds of basin features. We look, as she said, for these sort of layered features that are in these kind of U or V shapes underneath here that represent sort of a slice, you can imagine, through the earth where a river or a lake basin would have been at some time in the past. And in general, we don't always know quite the ages, but in general, the ones, when I'm going to show a couple of examples of these, if they're outlined in the purple, we think they're from the last ice age. And if they're outlined in this blue, I think they're from previous glacial cycles, previous times, hundreds of thousands of years ago when sea level was lower at different times in the past. So that's a little primer of I'm going to show you a couple of examples of what this kind of data looks like and how we used it to find the places that we wanted to collect the cores. And so I'll show three examples. One of them is from the Norton Sound, right here offshore of Nome, where we see small lakes and coastal plain environments. One is from out here on the outer shelf, at the edge in the marine terrestrial transition, where we think we see the Paleo-Yukon Delta when the Yukon River would have flowed all the way across the shelf here. And then I'll show you an example from this middle section, which we think is really sort of representative of most of the Bering land bridge and most of the shelf, where we have these broad shallow valleys with meandering channels, lots of oxbow lakes and thaw ponds. The other thing you can see, in addition to all the little yellow dots that represent the core locations on my map, I also have these kind of purplish blobs in here. And those are essentially locations where, in addition to the data that we collected on the Sikuliaq, I've looked through all of the data that's ever been collected by the Sikuliaq and the Coast Guard cutter Healy when they've been driving around on the shelf. And we've been able to identify hundreds of thousands of locations where there are these kinds of river channels and lake basins that really give us this idea that, as you can tell, there's a lot of purple blobs across that map there. So that means there's a lot of channels and lakes out here, which is one of the reasons why we're sort of saying we think there's a lot of these rivers and ponds and lakes out here on the shelf. So here's one example from offshore Nome. Again, you're looking at this black line here is the seafloor, and then we're really interested in this kind of these places where we see these U-shaped features with layered kind of striped looking sediment here. We go in and we collect a core in here. That's what this brown rectangle represents. Here's an example of what the top of some of those cores look like, where we get this marine sand on top and then this kind of nicely layered sand and silt in there, which is, as Sarah mentioned, representative of the terrestrial material. It has that kind of terrestrial plant material in it that really is the indicator that it's not deposited in a marine environment. What we see offshore of Nome is it's heavily eroded landscape, lots of coastal plain tundra, maybe older paleo valleys. This is an example from the mid-shelf in between St. Lawrence and St. Matthew Islands. Out here, what we see is something that looks a lot different. We see these much older, much larger, big, broad valleys. And then on top of that, we have little tiny, kind of relatively small, tiny channels and basins. And there's so many of them that oftentimes they kind of cut into and sort of chop up one another. And then we have this sort of brown marine layer material on top of that. And then these are a couple of examples of what the cores look like. Again, that's nicely layered terrestrial material. You can see some marine sand above some of those, but very similar. And this is one of the areas from the mid-shelf. Josh is going to talk a little bit more about the pollen data and some of what that's been telling us about from here. And then the last example that I was going to show, Ryan's going to talk about this at the end, but this is an example of what the data looks like from this Paleo-Yukon Delta on the outer shelf. So out here, we're actually in a fully marine environment, but this is a place where, as the schematic is showing, we think that the river extended all the way out to the shoreline out here and then has a river delta that would have been underwater. And that's what we think this orange deposit is in here. And then when we've collected cores into this, we can see a really nice sort of transition between lots of marine shells and into this kind of erosional surface where we get into more terrestrial material. You have lots of layered organic rich silt and clay in this delta unit. So lots of material and things for us to work with. So those are just a couple of examples, and I'll turn it over to my colleagues to tell you more about those different sites.
Okay, I think you guys can see my screen now.
Looks
so
good.
Great. My name is Beth Caissie. I work at U.S. Geological Survey in California. And I wanted to talk about a couple things. One thing is, while we were on the ship, like Sarah said, we opened up the cores and we also just took little samples from the cores as they were coming out of the seafloor because we wanted to know what we were collecting. And so we would take these samples and we would sieve them, or we would make little smear slides with them. And sometimes when we would sieve them, we would get a sieve full of something like wood. And so in this situation, we were like, this is definitely a terrestrial sample. In other situations, we would sieve them and we would get, these are foraminifera. This is a single-celled organism that lives in the ocean. It lives either in the sediments or in the water column. And so we knew that we had marine sediments in this situation. And sometimes we would, actually very often, we would have this situation where we would see pieces of wood. These are little moss leaves, and this is a foraminifera. And so we have a mix of both terrestrial and marine organisms all together in the same sample. This is another example of that. This is from a higher powered microscope, and we have a plant fragment here. And this is a stack of marine diatoms. So marine diatoms are single-celled algae. And we know that these are marine. So again, we have this freshwater marine mix, which was really confusing to us on the ship. So when we got off the ship, the first thing we did was we went to Oregon State where our cores are being stored, and we opened them up and we took a look at them. And we're going to use 3 different things in order to figure out what the marine terrestrial boundary is. The first thing is sedimentology. The second thing is isotopes, and the third thing is diatoms. So I'll go through each one of those. So by sedimentology, I just mean we looked at the cores. And so in these images, you have an image of the core on the left and a CAT scan of the core on the right. And you know, this is, I've sort of focused in on the boundary in each one of these images. And what you can see is that the marine sediments tend to be massive, very mixed sediments, sandy sediments often, sometimes a little finer grained. And the terrestrial sediments below tend to be layered. And there are 3 different kinds of layers. There are dark sand layers, lighter silt, and then peat layers, which is really exciting when we find these peat layers. Sarah talked a little bit about isotopes. And so basically what we did was we took the sediments that we had collected and we said, okay, where did this organic carbon come from? And so we used a couple of different things. I'm going to try to go through that a little bit. The first thing is carbon to nitrogen. And so this y-axis is showing you the carbon to nitrogen ratio within each one of these samples. And what you can see on the y-axis is there's a range from marine phytoplankton are lighter and land plants are heavier. We also did isotopes. And so just in case you don't remember what an isotope is, carbon has a few different isotopes. In this situation, we're looking at carbon-12, which is the most common form of carbon, where it's got 6 protons and 6 neutrons in its nucleus. Carbon-13 has an extra neutron, and so it's a little bit heavier. It's not as common as carbon-12, but it is naturally abundant. It's not radioactive. It's out there in the environment. And so we can actually use a mass spectrometer in order to figure out what the ratio is between these and come up with this thing that we call delta 13C. And that, again, ranges and it changes based on, that's what this x-axis is. Marine phytoplankton tend to be less negative than lake phytoplankton or land plants. And so you can plot all these, and each one of these dots is a different, each symbol is a different core. So this is basically all of the samples that we've taken. And you can see they sort of mix between this land plant box up here and this marine phytoplankton box down here. Soil is in the middle, so I'm not really sure. It's hard to sort of distinguish what's going on there. The third thing we looked at is diatoms. And these are just some examples of freshwater diatoms. They're outlined in green because each one of these images was taken from a terrestrial sediment. These are some examples of marine diatoms. The ones that are outlined in blue were taken from marine sediments. The ones that were outlined in green were taken from terrestrial sediments. So again, we have this weird mix, like we think we're in terrestrial sediments, but we're getting marine diatoms. What's going on? We put these 3 things together, and you can see the sedimentology here. You can see this big shift from terrestrial to marine sediments. These are diatom plots, and so each one of these pies in the blue, these are different types of marine diatoms. In the green are freshwater diatoms. So you can see at the top of this core, it's mostly marine diatoms. But then as you go down, it's mostly freshwater diatoms. But there's a lot of marine diatoms present. Up to almost 40% of the diatoms are marine, even as you get deep into this core down to 4 meters. We can look at the carbon to nitrogen ratios, and you can see that they're pretty consistently heavy until you get to the top of the core, and then they get light again. And so this shift happens, around 30 centimeters or so. We have the marine sediments at the top and the terrestrial sediments below, as evidenced by these three different things. So we came up with this sedimentation model, which basically we thought, like, how are we getting this mix of marine and terrestrial things in our sediments, even though we think we're in freshwater? And so the idea is, if you think way back, maybe 35,000, 40,000 years ago, the Bering Sea was like it is today. It was a big continental shelf, shallow continental shelf that was basically the ocean. There were whales, there were marine diatoms, there were walruses, there was sea ice. And so these marine diatoms are living in the water and settling to the seafloor. As sea level falls during the last glacial, the Bering land bridge is exposed, and we start to create the land bridge, but we're creating it actually out of this continental shelf. And so it's actually marine sediments initially. Rivers start to flow across those marine sediments. Soils are going to start to form, and you get these rivers that are sort of cutting through this old marine sediments and picking up these marine diatoms. As time goes on, the land bridge really develops. We start to get some wetlands. We start to get plants growing and things like that. We get lakes developing. We start to have freshwater diatoms living in the lakes, freshwater diatoms living in the rivers, and those are falling down and depositing in the sediments, but these rivers are still cutting through these marine sediments below. So now we have a mix of both freshwater and marine diatoms. Time continues, sea level is going to rise, and we're going to bury these ancient Bering land bridge basins. They're still there with their mix of freshwater and marine diatoms, but now we have marine diatoms being deposited on top. And there's not a lot of ways for freshwater diatoms to get in unless there's real huge winds or something like that. So now I am going to pass it along. Whoops. Josh, I think you're next.
Yep, I think so too. All right, so here we go. Let me share my screen. Okay, so my name's Josh Barna. I'm a PhD student here at UAF. My part of the project is to look at pollen and spores in the sediment cores. So the core that I'm working on right now is called Deep Blue Sea. This is where it was located or collected about 80 kilometers north of St. Matthew Island. It's 7.5 meters long, and it was from a water depth of about 70 meters. So to give you a little background, why do we use pollen for paleoclimate reconstruction? Well, pollen can persist in sediment for several thousand years, so it's good for a long-term climate record. It's sensitive to climate change and can be used to reflect temperature, moisture, and environmental changes. Pollen itself is widespread and abundant and produced in large quantities and is found in very diverse environments. It has a distinctive taxonomy, so plants are identifiable down to the family level and sometimes the genus level, which is useful for reconstructing past vegetation communities. It is useful for correlation with other proxies and complements other things like isotopes, ice cores, and sedimentary DNA, which Kira will talk about in just a few minutes. It's also useful for quantitative climate estimates. When you start to add it to climate models, you can use it to infer past temperature and precipitation. And then finally, it's good for ecosystem and human history, tracking human history and ecosystem changes. So you can see vegetation shifts, changes in megafauna habits, and their migration routes. So this is a close-up of the chirp plot for the score. This is kind of zoomed in on what Jenna was talking about. You can see here there is a transition between the marine and terrestrial sediment in this chirp plot. So below this red line is all terrestrial. And then there is an unconformity in our age model. Right here, you get into this lower, older stuff below that. It's still terrestrial, but it's a different isotope signature and sedimentology. And here on the right of my plot, is a photo of the sediment core that I was using. Above 450 centimeters is marine sediment, so I didn't really look at pollen in that because preservation is poor, and we're really looking at the terrestrial sediment for this analysis.
Josh, quickly, what is chirp data?
So this is the subbottom profile stuff. This is what Jenna was talking about. That's the terminology that was used. I don't think it's
So
an
it's the sonar or acoustics?
Yeah.
Okay. Thank you.
You're welcome. Sorry, there's a cat in the way. Oh. Okay, and so based on the isotopic signature, we were certain that this was terrestrial sediment in this gray box here on the right side of my screen. So this is kind of what this is exactly what I was looking at in my sediment core. You can see several different types of pollen. You have some woody taxa and some herbaceous taxa. There were fewer amounts of grass, or less grass than I thought there would be, which was interesting. There was a lot of sedge, mostly sedge, as you'll see in just a second, and surprisingly a lot more woody taxa like spruce, alder, and birch and willow. So here is the fruits of my labor after counting 350 pollen grains per horizon in this. I made this pollen diagram. So as you can see through time, there's little change, mostly sedge in this column here, a lot of grass. But what was surprising and also unexpected was the amount of woody taxa that we see, as indicated here in the green parts of this board, or this diagram, excuse me. And I think that's it. Sorry, that's kind of brief. So on to Kira.
Share my screen. Okay. Hi, everyone. Thanks for having us. My name is Kira Wankett, and I am a graduate student at the University of California, Santa Cruz. And what I do is I use sedimentary ancient DNA to characterize ice age environments, like Gay so wonderfully said at the beginning. So first I want to talk a little bit about what SEDA-DNA is. This is a concept that is foreign to many people for good reason. It's not very widely used yet, but SEDA-DNA stands for sedimentary ancient DNA, and it refers to these DNA molecules that we can find bound to sediment particles inside sediment cores. So here's my little rendering of what that looks like. These big brown blobs are sediment particles, and then these are the DNA bound to them. And we actually have pictures of what that looks like in real life. So this is work. These pictures are stills from a video produced with an an atomic forcing microscope by researchers at the University of Copenhagen. And you can kind of see it. The DNA is this kind of stringy dotted material here between these two pieces of mineral. And you can, if you squint and really look at it here in figure H, you can actually see the double helix of the DNA. So this really cool, really shows how the DNA can bind to the minerals, and the minerals will actually hold it there for thousands and maybe even millions of years. So that can form these really useful records that can go way back in time. Yeah, it's just really, really, really tight binding that I can then extract. So how do we use this sedimentary record of DNA to actually reconstruct landscapes? Well, here is a quick little rendering of what an ice age landscape may have looked like in Alaska, for example, 30,000 years ago. We have all these different plants and animals hanging out in the landscape, and they all have DNA. Everybody has DNA. And they're all shedding that DNA into their environment all the time. And when it rains, all of that DNA gets washed into the waterways and then collects at the bottom of these lakes. And so we can use lake cores to the lake cores really preserve this DNA record in a useful way for us. And so what I did for this project is I used this sedimentary ancient DNA record looking at two cores to start with from this expedition. The first one is Deep Blue Sea. That's the one that Josh has also been working on. And then also this Basinito site a little further south here. So the goal here has just been so far to actually make sure that we have DNA preserved in these cores, because not all cores preserve DNA. We don't really know why. It's kind of mysterious. So I took 2 samples from each of these 2 cores. And then I went into my lab, and here's a picture of what that looks like, all suited up in this full body Tyvek suit with a mask and 2 pairs of gloves. It's very involved. But I extracted DNA from these samples, and I found, I was able to characterize first the plant communities. So here I have everything. I'm reporting all of these species, all these taxa down to just the family level, just because otherwise my figure would be huge. And the number of reads here, these different colors, corresponds to how much of the DNA from that taxa I found in these samples. So the lighter colors is less DNA, and then down here to the dark blue is more DNA. And so I found some things that we expected. I found willow, which loves wet, marshy, environments. If you're getting lake cores, you're going to have willow in it. It's the rule of SETA DNA. We also found grasses and sedges. Also makes sense for a watery region. But something really interesting we found is spruce. So you might notice that a lot of these, there's a lot of overlap between the things that I found and the things that Josh found, which is a good thing, because that means that we're doing something right if we can both find the same taxa. So this was really exciting. But then even, I wouldn't say more exciting, depends on your perspective. I was very excited by the animal communities. So this is brand new data. I just made this figure today and just got all of this data back in the last couple of days, but we were able to find several different animal taxa in these cores. So I found ptarmigan up at the top, then mammoth, lemming, reindeer, bison, and geese, mostly Canada geese, which was pretty interesting. Can never get away from them. So I just put some pictures here of these animals and what they may have looked like on the landscape together. All of these guys hanging out. It's pretty cool to think about. So then we're really trying to get to that question of what did the habitats look like? So I clustered all of these different taxa into broader categories just to kind of get an overview of what the habitats were. And I found lots of shrubs and trees, this dark green bar here, and a varying amount of grass, kind of depending on where you are on the land bridge and when you are in time. And then I found very few flowers and herbs, just a little bit of this orange daisy here, that group. So that was very exciting. Looks pretty shrubby, not very dry and grass-like. So that was something that was really cool. I'm going to refrain from making any generalizations about the animal communities yet, because we just learned about that, and I don't want to say anything before we can all talk about it as a group. So that's all I have, and I think it's Ryan's turn.
Howdy, y'all. I'm going to be talking to us today about the northwesternmost transect that we performed along what is the Navarin Basin, this low spot on the Bering Shelf region. As we've seen a couple times before, plotted on the slide is the chirp subsurface transect, aligned from east on the right to west on the left. And I've color-coded several of the units that I've observed and that I'm focusing on for my PhD research. The lowermost unit that we're seeing that I have outlined in yellow, some of the carbon dates, or the carbon date that I've established from this point dates back to about 44,000 years ago, which is right during the glacial period. What's interesting about the termination of this section is that there's a unconformity. These sedimentary horizons are cut off. And you'll notice these like linear stalactite looking features at the top. Those are sea ice scour marks. So when sea level was lower and that surface ice was able to drag across the sediment and erode away the top part of that unit. Followed then by this red-brown unit here, where we have this long flat-lying strata with carbon dates coming back to around 15,000 years ago to 14,000 years ago. This is during the deglaciation, so as sea level was rising up. We're interpreting this as a delta deposit coming off of the central Bering land bridge. And then finally, at the very top of this is that I have outlined in blue is the modern marine snow. So the algae and the detritus that's coming out, it's falling down from the surface of the ocean, depositing itself here. And then you'll notice between those two units, it's a pretty gradational, like there isn't a large a boundary layer between those two. And so I'm really interested in the algae, the diatoms that are preserved in these cores. The northwestern part of the Bering Sea is oftentimes referred to as the green belt. It's an area of high productivity. There are a lot of this little diatom called Caissie living around. Just as Beth is performing, looking at the ratio between the marine diatoms preserved and the freshwater diatoms preserved in the sediment. And so how I gather this information, I take a unit of sediment from our cores and put it into a settling tank where the diatoms are dispersed evenly throughout the water. I let them settle out over the course of several hours where they fall on top of a microscope slide. Then I take all the water and remove it from the tanks. And then I have this dry layer, this dried out microscope slide, and I perform transects across. And I count every diatom that I encounter and I identify it to the species level. It takes a lot of time. I try to count about 300 or greater than 300 diatoms per sample. It takes about a day to 2 days per sample. And so I want to show you some of the data that I'm collecting from all of this project. So in this set of plots, we have core depth on the y-axis and a couple different variables plotted on the x-axis. Over on this orange plot, and this core site is at our very western edge, and we'll see that it goes through all three of those units, and I have them shaded at their corresponding layer in those colors. We have a high level of overall the diatoms preserved, and then there's this really dramatic drop as we're in this delta deposit, and then a resumption to higher diatom abundance. And for reference, this 10 to the 8th means that there's about 10 million diatoms per dry gram of sediment. So there's a lot of these little guys that were preserved overall. And then as we saw in Beth's presentation earlier, that carbon to nitrogen ratio, where we have a switch from near shore going to offshore. And what I'm showing here in this video is the CT scan, that CAT scan, that can help us reveal what's inside of our sediment cores. The CT scans do a great job at highlighting different densities inside of sediment. And so what we can't see from just the photograph can get revealed from looking at those CT scans. I'm going to play that one more time. So I applied a filter to remove the low density material. And then what we can see is I'm going to pause that really quick. There's a little bivalve shell that was in life position where its feeding tube would have been extended vertically. And there's a really sharp contact at the bottom of this red unit, that delta. And then as we get towards the top, we see that it's pretty gradational overall. So it was a smooth transition between the modern ecosystem that we know today. And then I want to show off some data as well from closer to the Bering Shelf, where we only have 2 units preserved, where we have this transition from a nearshore system all the way to our offshore. And this core overall was 13 meters long and presented from the very top here to the very bottom and the bottom right. And what we don't see in this core is a sharp contact where these 2 units would meet. There isn't a very dramatic contact point between those two. It graded and was moving along. And so going forward, I'm going to be projecting how this delta deposit was positioned through time, or it's through space, rather, using the chirp profiles from along the south, further to the south, and using the sediment cores from those transects to establish how there could be differences in that primary productivity, the diatoms. Lastly, I'm leaving us all with this and how we are reinterpreting the Bering Land Bridge, with previous literature suggesting that it was a grassland, and from our new data, we're reinterpreting it as it was much more likely a pretty boggy space. And with that, I think we're all taking questions.
And I just want to say, to answer the question raised by the entire presentation, in terms of was it, great grazing or buggy bogs, there were probably both at different places. But what we have mostly been looking at are the low-lying areas of the Bering Land Bridge, the gently sloping areas that were floodplains of rivers draining Alaska and possibly even Siberia. And so we think large areas of it where we're finding all those little lakes were significantly wetter and maybe shrubbier than Alaska or Siberia, which were covered by these arid grasslands.
All right, so thank you all very much. That was a lot of information to take in. And at this time for Strait Science, this is when we ask the audience to thank you all for your work that you're doing and your presentations and taking time to give us information. And we try to show a little love in the chat box because it's never easy to be a public speaker. And we very much appreciate your time bringing all this. There's quite a bit of information there. And I have questions, but before I get going, we'll throw it open for, you could throw your questions in the chat or unmute yourself using the unmute, click on the microphone and the slash button will come green. And you can ask your questions as well as video, undo the video as well. So we thank you again. Rick Toman, I see you've got your hand up. Go ahead.
Yeah, hi. Thanks, Gay. Thanks, everyone. Really interesting presentation. I had a question, and maybe one of you said this and I just missed it. But in your work here, were you able to differentiate areas that, comparatively speaking, rapidly transitioned from terrestrial to marine versus a somewhat longer drawn out process? Thank you.
I can answer that. And the answer is absolutely yes. There are some sites where we have a really sharp, we look at those CAT scans and there's a sharp boundary and you can probably there's time missing there where things were eroded away. And then there are others where you really can't figure out where to put that boundary at all. And so I think that was a slower transition. And we have a new postdoc starting who's really interested in looking at those transitions and is hoping to look at them in real high resolution, centimeter scale or sub-centimeter scale to figure out can we actually reconstruct water depth or anything like that? And so that's part of the ongoing work. But yeah, there's definitely both situations happening.
Thank you.
Great. I have a question, and that was on those where the sound is coming down from the ship and pinging down to the bottom like a big old fish finder. It was about 45, it could read about 50 feet down. Is that right?
We typically get images about 50 meters down, so 150 feet below the surface. So you can
15 meters?
50
meters.
50 meters.
Okay, I was trying to look at the scale that was on those sub-profile.
That, yeah, it just
It was
depends
like 5
on
meters.
it depends on the type of basically what the geology is. So if you have really fine grained mud,
Okay.
then you can get much deeper beneath the surface. But if you have a lot of sand, which we do, then or gas, then you don't go as deep.
Okay, so what I heard when I was seeing it, maybe I'm confused too, but for around St. Lawrence Island, you said there were so many canyons and, just really lots of um, canyons and disturbance or something, right?
Yeah, so in the most of the mid shelf and around St. Lawrence and St. Matthews and in that area, what we see is actually old river valleys from not the last ice age, but from previous glaciations. And so lots and lots of old river valleys where you had deeper rivers. And what we see and then what we see kind of carved in top, those are filled in. And then on top of that, we see lots of small channels and basins. And a lot of it really looks a lot like the picture. I sort of imagine it a lot like the picture in the upper right here. And all of those channels are coming in and they're cutting into one another and then they're erasing and they make it very, very complicated to pick out individual basins or individual channels, and you end up with lakes that are, oxbow lakes and things that is channels cut off, and it's sort of channels and drainage and lakes all over the place.
So then when Ryan showed I don't know if he's at a different scale, but he had ice scouring. So was Ryan, was your scale different on than the
Yes.
other way? Because then it looked like your ice scour was much deeper. And I was thinking, okay, well, so I had the scale wrong. I was looking at the 5 meter, it looked like was about a third of that profile.
So those images that Ryan was showing are highly vertically exaggerated. Like they're really, really squished. And so they are at a different scale than all of the other ones that we were showing. True kind of other ways that we didn't squish them all, if we didn't squish them up like that, it would be like this long, boring, flat thing where you wouldn't really be able to, wouldn't really see much. So we sort of squished it together and it makes it much more dramatic.
That's helpful because I was like, well, couldn't that all be ice scour? Because we just had that, we were talking about the cable earlier. The cable was in 100 and something feet of water up in off Barrow and or Licktock Point, and then that got cut by ice scour, and that's pretty deep. That cable was buried, and that's at 100 and something feet plus, moving that out. So it was just confusing. Thank you for explaining that.
Yeah.
Difference. It sounds like the marine sediment post the land bridge is how thick all that debris, just regular ocean debris that's piled up on top of the land bridge. How thick is that? Because
It really depends
looking
on
at
where
one scale, it was like maybe not very much.
It varies
it really
a
depends
lot
on where you
from site
are.
to site. So we have cores with 20 centimeters of marine sediment on top to some with maybe 4 or 5 meters. Beth, Jenna, correct me if I'm wrong, but those are.
No, you're right.
So like about 15 feet of just normal dust from above, just all our flotsam and jetsam debris from all our creatures and phytoplankton blooms and things like that. So 15 feet down in some places.
In some places. In other places, it's just a few inches.
Wow, from the original land bridge.
Yep. Because if you think about the Bering Sea today, there's a lot going on. There's places where stuff is just settling. There's places where, you know, the currents are really ripping, and so they're actually picking stuff up actively, and there's erosion happening and everything in between.
All right. Well, thank you. That's very helpful. That's great.
Question.
Go ahead, Edwin. Edwin?
Yeah. At what time frame was the ice thick enough that you know they were talking about the scouring to the ground in that area, and then at what time frame was it like really frozen to the ground?
I don't think.
Oh, go ahead, Beth.
No, you can go.
What was I going to say? So those ice scours are from probably 30 to 40,000 years ago, but it's important to remember they're deep now, but the shoreline would have been much closer to where those ice scours are today. So they're actually really close to the beach, and then they would have been in much shallower water at the time that the ice was touching the seafloor then. So I'm trying to remember exactly how thick they would be, but it's not as dramatic as having, like, you know, 100-meter thick ice or something like that. It would have been in much shallower water at the time that sea ice was scouring the seafloor.
So around that time, the level of the ocean was like really way down there.
Yes. Yeah, so exactly. Sea level would have been a lot lower, and the whole, you know, this is when the whole land bridge would be exposed. Your shoreline and your beach would be much further to the west, and then your ice would be offshore even further to the west there. But we do see that kind of sea ice scouring. I mean, you can see modern sea ice scours sometimes in those sonar images, and we, you know, see various sort of, mostly, you know, you're only going to see it in the marine environment, so you have to have, you know, sea level flooded, and you can see those kinds of sea ice scours in there. But
does that answer your question, Edwin?
Yes, that's very interesting.
Yeah.
Thank you.
And I was going to follow up and say, do you know how low sea level was from your work during the land bridge? So like where we are right now, how low did it go? How low was did the water
From
go?
global proxies, about 130 meters.
Yeah, and that's not
130 meters
from our work.
it's
I
not
mean,
us.
our work is consistent with that, but we were already sea level estimates from previous work. So yes, that's correct.
But the cool thing that we found is that we saw these notches that look like they're beach cut. Jenna, what's the word?
paleo shorelines, yeah.
Yeah, like these paleo shorelines. And up near where Ryan was talking about, they were at 120 meters. And then in the southeastern Bering Sea, they're a little shallower, like 100 or 110 meters. So it's like, that's definitely work we have to figure out and work on. But it's kind of exciting that we could actually see that.
So maybe we were like between 300 to 600 feet. The sea level was 300 to 600 feet lower. Something like that. Is that what I'm hearing?
No, like
No.
300 to 400.
Okay, 300 to 400 feet. All right. Wow. Thank you. That's very interesting. All right. Any more questions in the chat? Looks like you guys are written a book on, what is it called? The Dynamic Bering Sea, says Rick. All right. Any other questions from the audience? I got a million. This is the land bridge. I've got one for Kira. You did a great presentation on your animals and plants. And so it looked like there were willows, and then you showed the animals, and then, you know, I couldn't read the time scale on there. It's like KA, and I'd have to look that up what that was. But what's the time scale for all that biology that you were seeing on the land bridge?
So three of
Roughly, I mean, it's probably like thousands of years from when you saw it to when it started and stopped, but
So three of those samples, according to the dates we have now, are about 17,000 years old. And then another one's about 14,000 years old. So those are, I don't know how many years the samples themselves represent. Kind of depends on the site, but not that much time is represented in one sample. But they're all about 17,000 years ago.
Wow. Okay. So I'm going to ask a selfish question then. And that was one time I was in a trawl survey for king crab in eastern Norton Sound, and we hung the entire boat up and we'd hooked on with a net to something that was very large. And what we had to back down and get, figure our way out. And when we got the net back, there was a pristine, no growth on it, no debris on it, a mastodon molar. So that had to be kind of something that was, I think we hung up probably on a tusk or a big portion of the skull. And this was like 6 miles offshore of maybe St. Michael. And what's so at this point, how that would be thousands of years old, but the water would have been way down below. So that would have just been all grassy plain where that mammoth was walking. And of course, people find things in the Nome region. So,
or possibly somewhat shrubby. Mammoths' diet are a little bit more varied than some of the animals that are pretty much exclusively grass eaters like horses. And interestingly, Kira did not yet find horses.
Yeah, none of those samples had horses, just the things that can eat plants other than grass.
So you guys have taken a huge set of cores, it looks like, right? Norton Sound, St. Lawrence Island, down to St. Matthew, down to the southeast Bering Sea. So are you going to, hopefully you'll come back and tell us the rest of the story. Are you guys going to analyze like you're doing all those cores?
We hope to get to a core from all of our sites, although often we have multiple cores at each of those sites. So probably not going to get all of those opened and analyzed, but one of the reasons we're going back in May to open some more cores is we are trying to get our best core from each site.
Okay.
I have another question on
Go for it, Edwin.
did you guys come across any findings, like if there was any, like if a lot of water was pushed up this way from down south, and any high level of water that may have pushed up this way? No?
From south to north?
Yeah.
Okay.
Like if there's any would there be any tsunami that would happen in the shallow waters, or?
We haven't seen anything in the cores that I thought looked particularly like a tsunami layer, although it wasn't something we were really looking for, but that's a really interesting question.
There is a really old core that was taken in the middle of Norton Sound, and it has a bunch of storm deposits in it. And the thing that's pretty cool about it is that the storm deposits are really thick at the bottom of the core, and they get thinner and thinner as you go up. And so the idea is as sea level rose, the shoreline got farther away, and the storms would sort of come in and into Norton Sound and hit the shores to the east, and then come back and bring all this sand into this core site and like deposit all this sand. And then as the shoreline got farther and farther away, as sea level rose, those storm deposits got less and less. But that's something old that somebody else took that core and talked about that. But that's all I can think of that would be sort of relevant to that question. But it's really cool.
Austin, I see your hand is up. Thanks, Edwin. Sorry, Austin, did you get your question answered there, Edwin?
Yes, it's thank you guys.
All right. Austin, go ahead.
Yeah, hey, thanks. Great presentation. So, this really challenges, I guess, assumptions about what the Bering Land Bridge looked like. And I guess a good question that I have is like how, like the next phase, I suppose, might be, or the next question might be, how like animal migrations or human migrations may have been influenced by our new understanding of what it looked like. You have any opinion about that?
That is one of the questions we wanted to investigate because, of course, some animals did not cross the Bering Land Bridge. And there have been some questions as to why that might be, like did woolly rhinos never make it from the Russian Far East to Alaska? And one of the reasons really might be is that some of this habitat was difficult to travel through. It's, I think, a little bit more difficult to travel through a muddy, potentially boggy or lake-dotted floodplain. And it also may be that it was more shrubby and the food that those animals weren't looking for wasn't available, or wasn't available at the point where they encountered the land bridge. Certainly things like horses and mammoths, some grazers did cross, so there must have been places where there was great grazing. But there were also places where it was probably poorly drained, muddy, lots of lakes and potentially bogs. And if an animal, that's looking for good grazing encounters the land bridge, gets to the margin of the land bridge in one of these wetter floodplain areas, that might be why they didn't cross. And with respect to human migrations, one of the things we were really looking for is simply what kind of resources might have been available to people coming across the Bering Land Bridge. So we're looking forward to getting more pollen data across the region that we've sampled to look at differences from south to north. But right away, we are finding maybe more woody plants, shrubs, and even trees than previously would have been expected. So it does look like woody plants would have been a resource available to humans.
That answer it, Austin?
Yeah, maybe a follow-up, I guess, I think the Bering Land Bridge migration concept is getting slowly debunked. Do you think this sort of debunks is another one of those areas of study that kind of debunks that?
Do you mean the migration of human populations?
Yeah, because it the Bering Land Bridge migration corridor can't account for pre-Clovis culture. It can't account for the Mount Verde site in Chile or other much older sites. Yeah. That's what I was thinking about.
If you're talking about going through a corridor between ice sheets sort of in Alaska and Canada, yeah, that's problematic. But I think a coastal migration route is an alternate hypothesis that would allow people to move through the area earlier than an ice-free corridor.
Exactly. Yeah.
And
That's
so
what
I think
we-
we are really wanting to look at the vegetation a little farther south on the Bering Land Bridge, because that's interesting to know if it was a coastal migration, what resources were available there. And we've got sites a little bit further north right now.
Yeah, I've been arguing that our people have been mariners for a very long time, and we didn't need the land bridge to get here. Yeah. Hey, thank you. Really good presentation. Thank you.
Thanks, Austin. Yeah, were there no in the biology section, was there no human remains with the DNA? Would there be if there were people? And
we are not currently looking for human DNA. You can look for other DNA, and Kira, do you want to explain? You can mask any human DNA?
Yeah, so I didn't look for human DNA. We can mask it. It's also very low abundance in the cores, so that's not a very difficult thing to do to mask it.
What does the masking mean?
That means that we can hide it. So if it's there, I don't see it.
And we are interested in looking for that, but we want to talk to tribes, and we are currently preparing an inventory of the cores that we think might contain human DNA. It would only be snippets. It would never be enough to reconstruct a human genome, but it would tell you presence, absence of humans potentially if you found it. So we're preparing an inventory of those cores. We have a statement of how the method works and why we're interested in looking for human DNA, but we want to talk to the tribes that are bordering the Bering Sea about this, both for the inventory because there may be evidence of humans, and to see if they are interested in having us do that. So we'll be shortly sending out an invitation to the different tribes to collaborate on this draft inventorying the samples that may have human DNA, and seeking interest and permission to look for human DNA.
Oh, like test people and then match it up with what you have kind of thing?
No.
No.
Not groups
Look
of
at what
people.
you have.
There are characteristics that would say this is human, but you would
Okay.
never be able to figure out which group of people.
Okay.
Yeah, the DNA is not that specific.
But presence or absence would be pretty interesting, I think, for your time scale there.
We think it would, but we want to talk to the tribes because it's their ancestors that we would be looking for the presence of.
Yeah, very good. Any other questions? Mary Jane, I was thinking of you when they did the plants. I don't know if you have any question on the plants. No question.
Can I just say one other thing to Austin? Is
Oh
there
no,
was-
Mary Jane, you were unmuted.
Oh,
Go ahead.
she's talking. Go ahead.
We cannot hear you. I do not know why. You are unmuted, but I cannot hear you. It's just silent. Sorry to say. You could type it. For some reason, sometimes these things don't work, these Zooms. So you could type, if you have a question, just if you hit the chat bubble, which is down on the bottom bar, looks like a little cartoon. It says chat. Click on that and you can type in your question on typically the right-hand side of the screen. If you have a question on that for these guys. The last thing I had a question on while Mary Jane may be figuring on that is you had a question like why you had this marine and you had mixed marine and land. What mixed marine and land
Like
bits
freshwater and marine sediments mixed together.
So would that have been, if the Bering Sea, if the Bering Strait is opening up, right? I think there's a thought that it opens up, right? And then it starts filling in, or did it just sort of rise up? I don't know, but it seems like go ahead.
It was flooded. So like what does that mean, right? I mean, and so
did it come from something cutting through because the Chukchi Sea was high and or vice versa? Or was it just that the land finally came up with the water came up when the climate?
It's the water came up as sea level rose. So enough glaciers on land melted and put water into the ocean. So the oceans globally all raised up. Yeah. But then you think about like, how did that flooding happen? And it's an interesting thing to sort of contemplate. And I'm sort of imagining it probably happened the way flooding is happening today in Western Alaska, where, there's flat lying tundra areas that just as storms happen, they get cut off and, like things fall into the ocean. So, like at that moment that you have this transition and flooding is happening and you're going from the land bridge to the marine sediments, you would totally expect there to be a mix, or you'd expect there to be brackish, maybe like things that you'd find in an estuary or something. But the thing is that we find this mix all the way through. So not just at that transition, but all the way through. And so they have to be coming from somewhere else. And so that's why I think, we have these old marine sediments that the rivers are cutting through and picking up and bringing that stuff in there. Does that make sense?
Yeah, it does. And that's helpful. I really appreciate you guys spending the time to answer our questions. So Edwin had a good question about tsunamis, which got me thinking because we've had a recent spate of earthquakes in the Bering Strait right now, and that's been unusual, at least to me. Would the change as that water is either receding or the water's coming back up, would that have, and these glaciers are either melting or not, would that have created earthquake activity with the weight of that shifting so that you would have maybe a series of big waves either coming from the south, like Edwin, that was a good idea, or coming from the north if there's the earthquakes are coming there. So you could have a series of unrest, big waves. If that seems like a lot of weight, right? You're melting glaciers or you're bringing the ocean up or letting the ocean go down. Those are kind of big. I mean, big, right? Big scale. And I don't know if that's enough. I don't know. This is you guys are the experts. I don't know any of this, but would that be something that would have shook it up a little bit for your waves to be so mixed?
To
Your
generate a
results to be mixed?
tsunami, you probably need you need to kick the ocean floor. So most tsunamis would be coming from, for example, the subduction zone down by the Aleutian Islands.
Well, we're having a spate of earthquakes right now in the Bering Strait region, a 5 and a 4 and a.
Yes, and you can have them further inland, but they're usually not the kind that are going to generate a tsunami.
Yeah.
Okay. All right.
Because the fault movements are a little bit different than the kind required to give the ocean floor a little bit of a kick and set those waves in motion. But yeah, it's an interesting question whether, you might have some seismic activity, but.
Do you guys check with the peoples of the coast? And I say this because I think we do have historical stories of long ago of tsunami and waves that do hit this region. Are you guys checking? Because you guys have such interesting presentation and all these interesting findings. I mean, I'm so glad you're presenting to the Bering Strait region tonight. Are you guys checking with some of the documented kind of histories of the long histories of the peoples here to look and see if there's any information or what you're finding is correlating with stories that go back to when sea levels, there are stories here where sea level is different. Is that something you would look into?
Love to.
Yeah, because you have such interesting results. Does it match that?
Yeah, it would be cool to see if it matches.
Documented. What's documented? A lot of it was oral, but there is some that is documented about different events.
I think there's a little bit of a mismatch in timing because a lot of what we're looking at is, 22,000 years ago. We don't really know if there were people there then. And I feel like a lot of the recorded histories are going to be much younger. And so potentially in places that are inland, which is not to say that, I really think it would be amazing to, do some of those collaborations with people and learn what those stories are, because that would be really great. But I do wonder if there's a mismatch in time.
Yeah, it's certainly for our land bridge sediments, they're going to be too old. They're going to be older than those oral histories. But if we saw interesting features in some of the marine sediments, that would be a cool thing to do.
For sure.
Thank you. Thank you very much. All right. Any other questions? Thank you, Mary Jane, for putting in the chat that you didn't have a question. You were just really interested in the topic. So thank you. Yes, it was very interesting discussion. We thank all of you for your time. If there's no other questions, next.
Hold on one second. I think Beth had a follow-up for Austin. Did you want to say something else?
Yeah,
sorry
Austin,
about that.
I just wanted to follow up and say that there was a paper published recently that proposed that the human migration potentially used sea ice as a platform for both hunting and mobility. So, thinking about those people migrating along the coast, both in the summertime using, the ample marine resources that are there, but also in the wintertime, which to me just makes so much sense when you think about the people of Alaska today and how tied to ice they are. So anyway, I just wanted to mention that.
All right. Thank you, Beth. Sorry to cut you off there on that.
No worries.
Austin, any thoughts on that?
That's been my thought all along.
All right. Well, thank you all again. And maybe you guys can stay on for a sec. if you don't mind. I know some of you are in a different time zone. I really appreciate that. Next Strait Science will also be on a Wednesday next. That'll be March 20, actually, yeah, March 26th. And it will be the NOAA Polar Ecosystems Program, and they will be talking to us about the 2025 plans for the ice seal aerial survey, which will be coming out of Nome. I don't know if the plans have changed with the recent change to the government funding, but they are still, I haven't heard otherwise. So we think they are still on board coming in for, I think, a June survey, but they are going to talk about it and who they've been talking to about it and that sort of thing next week. So that'll be the Ice Seal Aerial Survey 2025 plans on a Wednesday night, March 26th. All right. With that, thank everybody. Thank you all for coming, and have a good night.